overall goal of this procedure is to isolate the 7 distinct silk-producing glands from the spider's abdomen and use these tissues for molecular and biochemical studies. This is accomplished by first anesthetizing the spider. The second step of the procedure is to separate the cephalothorax and the abdomen and pin the abdomen down with insect pins in a small dissection dish. The third step of the procedure is to make incisions in the exoskeleton to expose the silk-producing glands. The final step of the procedure is removal of the different silk-producing glands. Ultimately, results can be obtained that demonstrate that the different silk-producing glands express distinct silk mRNAs or proteins through molecular and biochemical approaches such as quantitative real-time PCR or mass spectrometry, respectively.
This method will provide important insight into spider biology research, revealing proteins that are stored within the silk-producing glands. These proteins have applications that affect the military, medicine, as well as many commercial and industrial applications. We first got the idea to do this particular study when we needed to learn more about the proteins that are stored within the silk-producing glands.
The main advantage of this technique is the female black widow spiders have very large abdomens, making it very easy to isolate the silk-producing glands. Individuals new to this method may have difficulty because oftentimes the glands rupture during dissection. There are also certain anatomical structures that are difficult to isolate. Visual demonstration is critical for a good dissection.
Demonstrating the procedure will be Coby Mattina, Tiffany Tuton-Blasingame, and Liang Zhao.